So this is just overview of digestion and absorptions of lipid. And in this figure, you can see the different enzymes uh, which is present in a different part of the GI system, and that is responsible for digestion of lipids. So you can see here this lingual lipids. So it is secreted from this ulnar gland of this dorsal ventral of the tongue. So this is responsible for digestion of short and medium chain fatty acid containing triacylglycerol. Similar to the gastric lipid, which is secreted from a chip cell of a stomach, and then it is also responsible for digestion of this triacylglycerol that containing short and medium chain fatty acids. And this pancreas, it secretes a pancreatic lipid, so it is the main enzyme that is responsible for digestion of triacylglycerol, so that contain in a food. So apart from that, there is a phospholipids, which is responsible for digestion of phospholipids, and cholesterol esterases, so that is responsible for digestion of cholesterol esters. So one thing here you have to remember that this in the previous session, that means on digestion of carbohydrate and digestion of a protein, we have seen that this carbohydrate and protein, they are completely digested. That means they are hydrolyzed into their monomers. That means this carbohydrate is hydrolyzed into the monosaccharides, and these proteins, they are hydrolyzed into the amino acids. But this digestion of lipid, that means the hydrolysis of these lipids is only partial. It is not a complete digestion. So this hydrolysis of carbohydrate and protein, it's a complete hydrolysis into its these monomers. And however, the digestions or hydrolysis of these dietary lipids, that is only a partial. It is not a complete digestion. So the whole session, so after the complete of this session, you will know about what are dietary lipids, and so how these dietary lipids are digested in our body. So that means which enzymes are secreted by a digestive system for the digest of these dietary lipids. And again, so what is the role of bile? So that this bile is also present in the small intestine, and it also aid on the digestion of a lipid. So how it uh, involved in the digestion of a lipid, so that also you will know. And after that, so what is the process? So from how these hydrolytic products of these lipids, they get absorbed from a lumen of intestine to the epithelial cell, that means in the sites. So, and also you will learn about what are the disorders, that abnormalities that may encounter in a lipid digestion. So dietary lipids. So that dietary lipids means those lipids which is present in a food. So in Indian foods and in Nepali foods, the average diet, it contains around 20 to 30 gram of a lipid. However, in case of a Western diet, it is twice to thrice time. So that means around 90 to 100 gram of a food, 100 gram of a lipid is present in their diet. So if we percentage, or that is these different components that present in a dietary food, that means generally in our typical diets, that it contains that 50 to 70% that is carbohydrate, and then there's 30 to 40% protein, and 20 to 30% that's lipid. But in case of the Western diet, that means it is fats, they also equally contributed. That means around is 50 to 60% of our diet, it contains a lipid. So these fats which is present in the diet, if it goes under specific lipids, then among that, this 90% of a fat which is present in the dietary food is only triacylglycerol. So 90 to 95% that fat which is present in the dietary food is a triacylglycerol. And apart from that, this in a smaller extent, that means in a fewer extent, there's other lipids also present in a dietary food, such as these phospholipids, and this cholesterol and cholesterol ester. And it's generally this cholesterol and cholesterol esters, they can be get from a animal source. And apart from that, others, these fats like this vitamin A, vitamin D, this vitamin E, vitamin K, and the carotenoids, they are also present in a food. So triacylglycerol. So triacylglycerol we can get from a animal source as well as a plant source. So on animal we can get this one from different sources like from milk, dairy product, butter, there's animal feed, fish, and egg. And depending upon the source, so from which source we are getting this triacylglycerol, the triacylglycerol may be the different. So the different on that means the fatty acid containing the fatty acid is terrified on that triacylglycerol. So these milk dairy products, so they contain a short and medium chain fatty acids, while there's animal meats, so they contain a long chain fatty acids on triacylglycerol. And apart from that, it's plant sources also we can get triacylglycerol. The differentiations on these plant sources triacylglycerol is this triacylglycerol contains more amount of this one unsaturated fatty acids than the saturated fatty acids on their this side chain of a triacylglycerol. So while we assume this triacylglycerol, so we have structures that means this glycerol, the three hydroxyls group of a glycerol is esterified with the three fatty acids to form a triacylglycerol. So triacylglycerol, three fatty acids esterified to the glycerol, and these three fatty acids they may be the different. So most of the triacylglycerols of a these are mixed type. So these triacylglycerols, then when we talk that means it's a two type. One is a simple and another one a mix. So simple means so all these three hydroxyl group of glycerol is esterified with the same type of fatty acids. And mixed type means that means there's different type of fatty acids esterified with a glycerol to form a triacylglycerol. And most of the triacylglycerol which is found in these dietary sources, that means from animal source or plant source, they are of a mixed type. So in addition to dietary lipids, the gastrointestinal system has to handle about okay. So apart from this triacylglycerol, it also contains this phospholipid and cholesterol ester. And phospholipids generally it is present in a cell membrane of a plant sources and as well as from animal sources. And cholesterol esters, they are from animal origin only. So it is not found in a plant source. So in addition to dietary lipids, the GI system has also this endogenous source might which should be handled during a digestion. So like this one example is so 1 gram of cholesterol and 5 gram of lecithin. So that enters intestine through while it's day. So these digestions of lipids, that is a more complex, considerably more complex as comparing to the digestions of carbohydrate and digestions of proteins. So since these lipids, so we know that it's a hydrophobic in nature. That means it is a water insoluble. However, the enzyme which is responsible for hydrolysis of these lipids, they are present in a aqueous medium. So enzyme is in an aqueous medium and the substrate is a hydrophobic in nature. That means uh, so which makes them a more complex. And next thing that this hydrolysis of lipid is also a partial. So that is there is a no complete digestion of a this dietary lipids takes place. So now I will discuss on one by one. That means the enzymatic digestions of a different substrate. So first one we'll discuss on enzymatic digestions of a triacylglycerol. So triacylglycerol is hydrolyzed by a group of enzyme called as a lipase. This triacylglycerol is hydrolyzed by a enzyme called lipase, and these different lipases are present in our body. So among them, that two of the lipases are present in stomach. So one is lingual lipase and another one is a gastric lipase. So lingual, while talking about this lingual lipase, so it is secreted by inner glands of a dorsal part of our tongue, so that it is secreted. So this lingual lipase, it is though it is synthesized in the mouth, but along with the food, it enters to the stomach. And next thing that the optimum pH for this lingual lipase is around 2 to 5. So that means it's an acidic pH. So this lingual lipase, though it is synthesized in a mouth, but it is active in a stomach. So though these but the enzymes which is responsible for digestion of this fat, so especially triglyceride, it is present in a mouth, but the actual digestion begins in a stomach due to this lingual lipase is active only in a acidic pH. So gastric lipase. So another lipase is also present in a stomach, and this gastric lipase is it is secreted via chip cells of a stomach, and the secretion is small quantities, and its optimum pH for the activity of this is 5.4, and it also required presence of calcium. And this gastric lipase is not much important in case of an adult, but it is more important in case of a infants. So in infants, this gastric lipase has a more importance because the substrate, so substrate which this gastric lipase it acts, it is majorly this lingual lipase or this gastric lipase, they are more active in the triglyceride which have a short chain or medium chain fatty acids. So the staple diet of a infants, that is a milk. So milk contains short and medium chain fatty acid containing triglyceride. So this gastric lipase, it's big, it is important in case of infants, that means they have a substrate. The substrate, the fat which consumed by infants is a triglyceride rich in a short chain and medium chain fatty acids. So the gastric lipase get a good substrate. Apart from that, the pH of infants, that is, it is not a acidic, so that's it's near to the this one alkaline, so that means around that 5.4, so that gastric lipase can hydrolyze well. So and another thing that already we have discussed in a previous slide, that one thing, these fats, they are hydrophobic in nature, and the enzymes, so these enzymes, they are hydrophilic in nature. So so that what happened, the hydrophobic means, that means when we keep for just if we keep a drop of oil on a water, then what happens, it's become a large molecules in a water. So similarly, these fats on a this lumen, they form a large molecules, so that it have a less surface size to expose, and so that the enzymes cannot act on that. So to solve this problem, these fats should get emulsified. So emulsification means that is the conversions of these large molecules into the small droplets. And so after the conversions of these large these fat molecules into the small these droplets, then it exposes large these surface areas, and so that these enzymes get acts on that substrate, and they can able to hydrolyze. And the emulsification, so it is helped by several factors. So one is this core body temperature. So the core body temperature of our body, this temperature of our body, it's make a liquidifications of fats, so that it's get soluble. And apart from that, this peristaltic movement of the stomach, so that is crunching of a stomach, so that also helps on a emulsifications, so conversion. So this, so for that we can assume one experiment. So that means if we keep a drop of oil on a water, and if we shake continuously, then what happens, this oil gets break down into the small molecules. So similarly, these peristaltic movements of a stomach, so that also help on a conversions of these large fat molecule into the small droplets. So apart from that, this lingual lipase and gastric lipase, they also aid on the emulsification. So by how means that in the beginning, the hydrolysis of these fats by lingual lipase and gastric lipase is very slow. So that means what happened, it, so after the hydrolysis by lingual lipase and gastric lipase to the triacylglycerol, they are converted to the diacylglycerol and fatty acids. And this product, that means the fatty acids, now it helps for the emulsifications of the remaining fats, so that the later on the hydrolysis of a fats goes speed. So initially it's a slow, and after after there's slow hydrolysis that produces a monoacylglycerol, and it aids the emulsifications further, and so that it speeds. So later on, that means what happened is lingual lipase and gastric lipase. It speeds the reactions. So apart from that, for the emulsifications, these dietary phospholipids and these free fatty acids and monoacylglycerol, they also help for the emulsifications. So these lingual lipase and gastric lipase have a specificity on a triacylglycerol. So they only hydrolyze a triacylglycerol that contain short chain and the medium chain fatty acids. They are able to hydrolyze the triacylglycerol that containing short chain and medium chain fatty acids. So the short chain fatty acids, after the hydrolysis of triacylglycerols by lingual lipase and gastric lipase in the stomach, the released short chain fatty acids, they diffuse through the stomach to enter the circulatory system. So this gastric lipase is particularly important in a newborn. So since at this state of life, pancreatic secretions contain relatively little lipase, and apart from that, the substrate is a milk, so which contains short chain and the medium chain fatty acid containing triacylglycerol. So apart from that, this gastric lipase, it also account for a 10 to 30% of hydrolysis of triacylglycerols that containing long chain fatty acids. Though the for the good substrate for this gastric lipase and the lingual lipase is triacylglycerol containing short chain and medium chain fatty acids. This gastric lipase is also able to hydrolyze the triacylglycerol, which contain a long chain fatty acids, but its contributions on this hydrolysis of triacylglycerol containing long chain fatty acids is very small. So that is around 10 to 30%. And while talking about the lingual lipase, the the contributions of this enzyme in a fat digestion is quite low. However, particularly it is important since it is a acidic. It's active. It cannot be inactivated by acid, and due to that, in absence of a pancreatic secretions of bicarbonates, this enzyme is in the small intestine as well. So at that time, what happened? This enzyme is able to hydrolyze triacylglycerols. So this lingual lipase is important in case of the patient or persons who is suffering from this pancreatic insufficiency. So that means unable to secretions of pancreatic lipase. On that, what happened? On those patients, this lingual lipase, it is active even in a small intestine and able to hydrolyze the fat or triacylglycerol. And the hydrolysis of this lingual lipase and gastric lipase is only partial. So they are able to hydrolyze the triacylglycerol into the fatty acid and diacylglycerol. So these are not able to hydrolyze, that the complete hydrolyze. That means triacylglycerol to the free glycerol and the free fatty acid. So that is not possible. So they only hydrolyze the partial, so that they are able to convert this triacylglycerol to the free fatty acids and diacylglycerol. However, this lipolysis by this lingual lipase and gastric lipase is important since the long chain fatty acids, which is produced by hydrolysis, partial hydrolysis of triacylglycerol in stomach, this after entering to the duodenum, these long chain fatty acids, they can stimulate the secretion of cholecystokinin, and in turn, this cholecystokinin it stimulate the release of pancreatic juice from a pancreas, and which contains this pancreatic lipase and these bicarbonates. Apart from that, the long chain fatty acids, they also have a antibacterial activity. And next thing, that diacylglycerol, which is the product of a hydrolysis by this gastric lipase and the lingual lipase, is a better substrate for a pancreatic lipase. And these formations of this monoacylglycerol aid of a fat absorptions. So it's help on fat absorption. Digestion by pancreatic lipase. So this pancreatic lipase is the main enzyme of digestion of triacylglycerol in the body. So the major part, the major is the digestion of a triacylglycerol that is takes place in a duodenum by a pancreatic lipase, and this pancreatic lipase is secreted by a pancreas, and for the activity it requires optimum pH that is a neutral, so it's around 7, and this environment, that is the pH environment, is provided by a bicarbonate ion, so which is present in a pancreatic juice and also secreted by a pancreas. So the emulsifications required for the activity of this pancreatic lipase to the its substrate, that triacylglycerol, it is further helped by a bile salt. So this bile salt is, these bile salts are produced in a liver, and they are released in a small intestine along with a bile. And this bile, the physical properties of a bile salts that enable them more active emulsifier. So these bile salts are more amphipathic in nature, so that they help for this for emulsifications of a fat. So that means conversions of large fat molecules into the smaller droplets, so that it reduces the surface tensions and exposes the more surface areas where these enzymes can bind to its substrate for hydrolysis. So these bile salts that containing 3 hydroxyl groups, example that cholic acid, are better emulsifier than those that only contain 2 hydroxyl groups, like this deoxycholic acids. So those contains more hydroxyl groups, they are more hydrophilic in nature, and they are the good emulsifier for a fat. And apart from these emulsifications by bile salt, these bile salts are also necessary for the absorptions of dietary lipids, and it helps in absorptions of dietary lipid. So this pancreatic lipase preferentially its attack on first and third bond in a triacylglycerol to produce a monoacylglycerol. So generally in triacylglycerol it contains 3 ester bonds. So three ester bonds where these fat acids are esterified with glycerols. Among that, carbon number 1 and carbon number 3, these 2 sites are preferentially attacked by a pancreatic lipase. However, on the second carbon, it is not able to attack. So that's why the product is 2 free fatty acids and a 2 monoacyl glycerol. So the product is 2 monoacyl glycerol and free fatty acid. So, and this pancreatic lipase, it is secreted in the active form, but for its activity, so for the binding of this lipase to its substrate, that is a triacylglycerols, it required another cofactor called as a colipase. So it's a small protein which is secreted by a pancreas, and so for the activity of this pancreatic lipase, colipase is necessary as a cofactor. So which is a small protein, and the main function of this colipase is it reduces the surface tensions at the liquid aqueous interface, so facilitating the interactions between the lipase and the lipid droplets. So generally this colipase, it's bind onto the liquid aqueous interface, and so this helps the binding of pancreatic lipase, so it is a hydrophilic in nature, to the triacylglycerol, which is hydrophobic in nature. So this is a hydrophilic enzyme, hydrophobic substrate. So for the interactions between these two hydrophilic and hydrophobic molecules, which is held by the colipase, so it's become a mediator for the binding of this lipase to the hydrophobic substrate, that is triacylglycerol. So another thing you have to remember is that is pancreatic lipase, it cleaves the bond presence in the carbon number 1 and 3 only. However, it is not able to cleave the ester bond presence in the carbon number 2 of a triacylglycerol. So the product is 2 monoacylglycerol and free fatty acid. And this monoacyl glycerol, so it is hydrolyzed by the another enzyme called as a pancreatic esterase. And this pancreatic esterase is a non-specific triacyl glycerol. It is also it can also hydrolyze the other substrate like these cholesterol esters and these phospholipids. But the activity of this pancreatic esterase is very low. So that's why only 25% of ingested triacyl glycerol is completely hydrolyzed to the glycerol and fatty acid before absorption. So otherwise the major products of a triacyl glycerol is that is the 2 monoacyl glycerol, 1 monoacyl glycerol. So and so the major is a product hydrolyzed by the major product after the hydrolysis of triacyl glycerol is the 2 monoacyl glycerol. So because this pancreatic esterase has a very low activity, so around only 25% of triacyl glycerols which is present in the dietary food is completely hydrolyzed into the glycerol and the free fatty acids. So otherwise, this pancreatic lipase, which is the main enzymes responsible for digestion of triacylglycerol. So which only cleave the bond between the first and third carbon of a triacylglycerol to produce the 2 monoacylglycerol. So that's why 2 monoacylglycerol is the major product after the hydrolysis of triacylglycerol. So now next one is that is a digestion of cholesterol ester. So this digestion of cholesterol ester, it takes place in a small intestine. This cholesterol ester, which is present in a food, is directly transported to the small intestine, where the specific enzyme responsible to hydrolyze this cholesterol ester is present. And the specific enzyme which is responsible for degradations of cholesterol ester is cholesterol esterase, and this cholesterol esterase is secreted by a pancreas along with a pancreatic lipase. And this cholesterol esterase, it converts cholesterol ester to the free cholesterol. So welcome back. So we are in a digestion of cholesterol ester. So these cholesterol esters, which is present in a food, they are directly transported to the small intestine. Since the enzyme which is responsible for digestion of this cholesterol ester is present in a small intestine. So the enzyme which is specific for digestion of cholesterol ester is, that is the cholesterol esterase, and this cholesterol esterase is secreted by a pancreas and released into the small intestine. So this enzymes, it cleaves cholesterol ester into the free cholesterol and a fatty acid. So similarly, these phospholipids and phosphatidylcholine. So these are also they are also directly transported to the small intestine. So the enzyme which is responsible for digestion of this substrate is also present in the small intestine, and this phospholipid A2, so which is specific for a hydrolysis of this phospholipid. So it is secreted by a pancreas along with this one cholesterol ester and pancreatic lipase. So the digestion of this phospholipids takes place in a small intestine. So this phospholipase A2, it's cleaved this phospholipid into the lysophospholipid and fatty acid. So here this phospholipase A2 is specific for a breakdown of this ester bond that is present on a second carbon of phospholipid to form a lysophospholipid and fatty acid. So pancreatic lipase, it's cleave a first and third this ester bond form in the first and third carbon, and this phospholipase A2, it's cleave phospholipids on the second carbon. So the ester bond presents in a second carbon of phospholipid to form a lysophospholipid and fatty acid. So after these digestions by this pancreatic lipase and this phospholipase A2 and cholesterol esterase, the product is that is the 2 monoacyl glycerol, free fatty acid, cholesterol, and lysophospholipids, and some this free glycerol as well. So does form these products. So they are now absorbed. So for the absorptions, the free glycerol, so which is produced during a digestion of a dietary fats. So they are directly passed to the portal vein and taken to the liver. So for the these fatty acids, the absorptions is differ according to the carbon content. So those fatty acids which contain short and medium chain, so that means less than 12 carbon, so that type of fatty acids are also directly passed to the portal vein. However, these long chain fatty acids, so that means more than carbon 12. So they are hydrophobic in nature, and so that they cannot directly pass to the portal vein. So this type of fatty acids, they are absorbed to the lymphatic system, and so and not directly to the blood. So this absorption mechanism is that's proposed by a Bergstrom, so which is the most acceptable theory for absorptions of these long chain fatty acids and partially digested products. So like this cholesterol, and then this two monocyl glycerols. So how this is absorbed and transported to the circulations is proposed by a Bergstrom. So the Bergstrom theory, so it includes three things. So one is formation of mixed micelles. So resynthesis of triacylglycerol in epithelial cell of intestine. And after that, so there is a transport of this triacylglycerol to the circulations by forming chylomicron. So first one, that is the formations of mixed micelle. So this mixed micelle is formed by the this different hydrolytic product of a digestion. So mainly this one two monoacyl glycerol, long chain fatty acids, cholesterol, so phospholipids and lysophospholipase. So these are incorporated with a bile salt to form a mixed micelles. And this mixed micelles is a very small droplets, so which is generally water soluble. And during formations of a mixed micelle, these hydrophobic parts, like this one, this two monoacyl glycerol, this long chain fatty acid, cholesterol, so they form a core of this mixed micelle, and it is covered by the amphipathic compounds like this one, phospholipids and bile salt. And this micellar formation, so this it is essential for the absorption of a fat soluble vitamins also. So these fat soluble vitamins, so they forms as a core part of this micelle. So the micelle aids the it helps the absorption of monoacyl glycerol and fatty acids by effectively increasing the solubility of this compound. And the micelle are aligned at the microvillus surface of the jejunum mucosa, so that is fatty acids, 2-monoacyl glycerol, and other digestive products, they passively diffuse into the mucosal cell, but not bile salt. So after the formations of mixed micelle, these fatty acids, 2-monoacyl glycerols, and other products, they become more soluble, and so that they can easily diffused to the epithelium. And for that, it is this for the divisions of these different products, the bile salt helps. And during these diffusions, bile salt is not absorbed on the jejunum site. This bile salt is reabsorbed by the another side, which is called as a another site, like in the ileum, and which is called as a enterohepatic circulation. So bile salts are mostly reabsorbed from the ileum, returned to the liver to be reexcreted, which is called as a enterohepatic circulations. So you can see in a figure. So from liver, these bile salts are synthesized and then they are released into the small intestine. So again from the ileum, that 95% of these bile salts are again uptake to the liver. So it is called as the intrahepatic circulation. And next thing is that the uptake of fatty acids into the enterocyte is facilitated by the presence of fatty acid binding protein in the cytosol. So the enterocyte, that's the epithelium of small intestine, it contains these fatty acid binding proteins. And after the absorb after the this small intestine epithelial cell absorb these long chain fatty acids, so they get bind with this fatty acid binding protein, and then this fatty acid is transported to the endoplasmic reticulum. So where these fatty acids get activated and again involved in the resynthesis of a triacylglycerol. So this is the summary of the this one of these different steps after digestion. So you can see in the figure. So this triacylglycerol, so which is present in the dietary food. So it is cleaved by a pancreatic lipase in presence of co-lipase into the 2-monoacylglycerol and these fatty acids. Now these fatty acids, free fatty acids and 2-monoacylglycerol with the help of the bile salts, it's form a mixed micelle, and this mixed micelle now it is transported to the this one jejunum part, mucosa of the jejunum. So from where these free fatty acids and 2-monoacylglycerol get absorbed, and after the absorptions, these free fatty acids they are bind with the fatty acid binding proteins and they are cleared from a cytoplasm. So this fatty acid binding protein it transported its free fatty acid to the after forming a complexons, it is transported to the endoplasmic reticulum. So where triacylglycerol synthesis takes place. And similar to this, some non-hydrolyzed triacylglycerol, so which contain a medium chain fatty acids, they are also absorbed intact into the cell of the intestinal mucosa, where they are hydrolyzed by a mucosal lipase. So in this epithelial cell, this lipase enzyme is also present there, so which converts this triacylglycerol to the free fatty acids and glycerol. So now next step, that's the resterifications of fatty acids and these 2-monoacylglycerol to form a triacylglycerol. So that is the resynthesis of a triacylglycerol. So it is takes place in a this epithelial cell of a small intestine. So these fatty acids, so which is transported to the endoplasmic reticulum. So they are activated by the anator enzyme acyl-CoA synthetase to form a fatty acyl-CoA. And this fatty acyl-CoA, they are esterified with a 2-monoacylglycerol to form a triacylglycerol. So which you can see in a figure also. So this one is that is the activations of fatty acid to form a fatty acyl-CoA. So after formations of this activated form of fatty acids, then it is esterified with a 2-monoacylglycerol to form a diacyl and diacylglycerol and finally triacylglycerol. So after formations of triacylglycerol, along with the other dietary products which is absorbed, like these phospholipids and cholesterol esters, these cholesterols, they form a chylomicron with the help of the protein called as a apoprotein B48. So after formations of this chylomicron, they are now released to the lymphatic systems and through the thoracic duct, they are released into the circulations. So free glycerol A from this hydrolysis of 1-monoacylglycerol and glycerol 3-phosphate from a glycolysis also serve as a precursors for triacylglycerol synthesis in enterocytes. So major substrate for the synthesis of triacylglycerol in enterocyte is that is the 2-monoacylglycerol. So which is the hydrolytic product by these digestions by the hydrolytic product of a triacylglycerol by lingual lipids. So, sorry, it's a pancreatic lipase. So pancreatic lipase cleave a triacylglycerol to the 2-monoacylglycerol and free fatty acids in a intestinal lumen. And this in the form of 2-monoacylglycerol and free fatty acids, they are absorbed to the enterocytes. And in the enterocytes, again, this 2-monoacylglycerol is esterified with these fatty acids to form a triacylglycerol. So apart from these free glycerols, which is held in the enterocytes by hydrolysis of 1-monoacylglycerol and glycerol 3-phosphates from a glycolysis, they also serve as a precursor for a triacylglycerol synthesis. However, the free glycerol held during a complete hydrolysis in a intestinal lumen is not utilized by a enterocyte for a synthesis of triacylglycerol. So the free glycerol, which is held by a complete hydrolysis in the intestinal lumen, these glycerol are directly transported to the portal circulations. So it is not available for a resynthesis of triacylglycerol in epithelial cell of a intestine. So this majority of triacylglycerol, cholesterol ester, and phospholipid molecules along with a apoprotein B48 and ApoA are incorporated into chylomicron. So this chylomicron is the carrier lipoproteins for a dietary lipids, and thus from these chylomicron are secreted to the lymphatic lacteal to reach the systemic circulation. So this is the figure how these chylomicron are circulated to the circulations. So chylomicron, it is formed in the intestinal epithelial cells by incorporating different these dietary lipids along with the apoprotein B48. They form a chylomicron. After formations of this chylomicron, now it enter to the lymph, and from the lymph it released to the circulations, and from circulations in the endotheliums of a blood that there is that another enzyme is present, that is the lipoprotein lipase, so which clips these chylomicrons, and from which these fatty acids are transported to the muscle and adipose tissue. So this figure represents the this is a short summary of digestions and resynthesis of these triacylglycerols, and finally circulations of these dietary lipids. So here, so after digestions by different enzymes in the intestinal lumen, they are converted to the different product like this: 2-monoacylglycerols, free fatty acids, cholesterol, free cholesterol, lysophospholipids, and these fat soluble vitamins. So they are get absorbed in the intestinal mucosal cell, and after reaching on the intestinal mucosal cell, these two by using these 2-monoacylglycerol and free fatty acids, that is again there is the synthesis of triglycerides takes place. So there is the resynthesis of a cholesterol, triglycerides, and phospholipids. And after that, all these dietary fats, they are incorporated to form a chylomicron. And for that, for the formations of chylomicron, the another protein particle cell, which is called as a apolipoprotein B48. So with the help of these apolipoproteins B48, these dietary lipids, they form a transport lipoprotein particle called as a chylomicron. And after formations of this chylomicron, it is enters to the lymphatic systems, and with the help of thoracic duct, this chylomicron is released into the circulations. From there, this fatty acids presence on that chylomicron is uptaken by a muscle and adipose tissue. So now, these abnormalities in absorptions of lipid. So when there is the abnormalities in digestions and absorptions of lipid is takes place, then there is the excess fat will be excreted in a stool, and so if there is the excess fat is present in a stool, then the condition is known as the steatorrhea. So steatorrhea in normal it is called as a fatty stool. So main cause of steatorrhea is that is the malabsorption or maldigestions of fat. So when the digestions of fat is altered, or when the absorptions of fat is altered, then this fat is excreted in a stool. So the various causes of maldigestions are, that is the decreased lipase activity. So if the pancreatic lipase, the activity of pancreatic lipase is low, then the dietary triacylglycerol cannot be hydrolyzed, so that this fat is excreted into the stool. And again, there is the, if there is any problem with the bile secretions or productions, also lead to the this fatty stool. So that also lead to the maldigestions. So this bile secretions, that it contains a bile salt, and this bile salt is necessary for the emulsifications and for the formations of mixed micelle. So mixed micelle. So this bile salt is necessary. So if any problem under these bile salt productions or secretions, or even the obstructions on the bile duct, there is a deficiency of bile salts. So in deficiency of bile salts, this fat cannot be digested and absorbed, so that it is excreted. Similarly, during a pancreatic insufficiency, so where these pancreatic enzymes are not synthesized properly, and this pancreatic insufficiency is generally seen in case of the carcinoma. And again, the use of lipase blocker drugs. So this, such for example, orlistat, so which inhibits the pancreatic lipase, so that is unable to digest the triacylglycerol presence in dietary foods, lead to the this steatorrhea. And again, the consumptions of indigestible fats like cholesterol, so lead to the steatorrhea. So apart from that, these are the various causes of these malabsorptions of a fat, like this inflammatory bowel disease, so celiac disease, beta lipoproteinemia, so which lead to the malabsorptions of a fat. So now this is how this steatorrhea is diagnosed. So the for the conformations of steatorrhea, generally this fecal fat excretions of 24 hours is measured. So the normal excretions of fat in 24 hours is, that is, less than 7 grams. So if the fat presence in the stool is more than 7 grams per day, then it is diagnosed as a steatorrhea. Okay, so these are the references from which this lecture is prepared. So thank you for your presence.